Yes, we are online. I can see it already on YouTube.
We are?
We are online, yeah?
Okay,
Yes.
because I somehow cannot see it.
No,
We're
it's
also
wonderful.
getting more people.
Yes.
6 viewers. Yeah, I see the viewers. Okay.
I wonder if it's just the 6 of us here, but checking on YouTube.
Okay, so shall we start then, since everything is set up? Great. Well, thank you everyone for being here today. Welcome to our September public seminar. We have some exciting updates about the network and the conference that was organized by our network. And also we have a keynote speaker for today, Sean McMahon from University of Edinburgh. I guess we will start with the conference.
Then take on this. So hello everyone, my name is Yannick. I'm here in Woodstock in Strasbourg, and I want to organize OoLEN's biannual conference or in-person conference once every 2 years. And this is happening right now. It's still on Monday. It's going until tomorrow lunchtime, roughly. And yeah, so as you can see, just from the little statue in the back, so I'm sitting here with Klaus Paschek, OoLEN board member, but also participant. And yeah, so you can see from the little statue in the back that we were lucky to got a fancy room in the Palais Universitaire. So that's the old university building. And it's, so far I can tell from me as an organizer, it's going quite well. Was a bit stressed out how things would work out. Normally, we should have been at least 2. I was organizing this with Omar Markovic and also Celia Blanco, who anyway dropped out before, but was a great help. Omer finally couldn't come in the end, but things are working well so far. We have a mixing program of scientific talks. So the people who joined, we are roughly 30 people, PhDs, postdocs, a couple of master students also, mix of scientific talks, but also discussion rounds on subjects like what are past achievements in the origins of life field and what would be future milestones we have to aim for, or um, um, things about biases in the origins of life research. That was a big thing. How to define life is a recurrent question. And yeah, and then even today, in the last discussion round, it got even a bit more fancier. So people started to write their own questions on the sheet. And there was the question about creationist arguments and even did we kill alien life? So this is just to give you a bit about the atmosphere that we have. It's pretty relaxed. We have a couple of people here who come for the first time to a conference, and the feedback I got from them was that they liked it a lot, that they could easily catch up with people, talk to people, and this is what we want. So yeah, so now at the moment, so people have gone to their hotels. We have a break before having a dinner afterwards in the city center. And tomorrow morning, we'll continue with one last session of talks and discussion rounds. So far from Strasbourg. Okay. Yes. So for that, now it's what wir Bildschirm freigeben. We'll get to where? Yeah, okay. Then we'll get on. So I have to go to the Habitanti. Wait a minute. I have to go here. It's Tuesday afternoon, and we have it. Still it's here. This is the organizational update. So just have to check whether we have here the full screen. Was Control L? Great. Yeah. Thank you so much. Okay, great. And now it's up to Klaus to give more details about OoLEN organization. Yes.
So everyone can hopefully properly understand me.
And I hope you can also see the slides. So yes, hello everyone. I will just give you a quick update, like what were the most recent developments in our wonderful Origins of Life network. And so first we had some changes to the executive board. So after a very long time of 5 years, Silke stepped down to be a board member, and she's like the last co-founder who was left on the board. And I just want to say a big thank you from everyone from the board, but also from the working groups and everyone in the network for her really great contribution, making this all possible and pushing OoLEN to hopefully now walk on its own feet to make a lot of amazing future conferences and so on. And then we have a new addition, which is Val, which is also here right now hosting, co-hosting the virtual meeting. And I want to say a big welcome from everyone, and we hope we have a nice and fruitful work together ahead of us in the executive board. Then I also, of course, a big shout out to everyone who is watching now or who is also later watching the recording. Of course, we would be super happy if you would like to put a bit more into if you want to go the next step and also contribute more to the network. Of course, you're always welcome to approach us. We can always need help, for example, in work groups. As one example, in the virtual meeting work group, we always look for people across the whole globe for all different time zones where we want to then host the meeting in different time zones. So everyone has a possibility to attend in their most comfortable time. And of course, you can also always look for people who might want to get involved in the executive board. Just approach us and let us know. Of course, this is your way to also maybe change some things that you have, or they are doing this very wrong and we should change this in a different way. Then of course, it's your chance to also shape OoLEN in the way that you might think is a good idea or how you would like to see it to evolve. So yes, please join us, and don't be afraid. The time commitment is usually a few hours per month. Let's say maybe 1 hour per week, something like in this kind of amount. So it's something that should hopefully be possible to just do while still managing of course all your academic life and so on and so forth. So we would be super happy for you to join us. Then just a short overview. Silke was actually so kind to provide some statistics of the recent growth of network, and you see we are getting stronger and stronger. We surpassed already now 200 members which are active in the network. And of course, if you have the chance, if you are at other conferences also, or other just talking to people who are interested in Origins of Life, of course, tell them that we exist and invite them to join us and tell them we have a lot of fun. And of course, we're always looking forward to have more people enriching our network. And this can literally be anyone who is like actively researching in Origins of Life field, can be a bachelor student, can go all the way up to even early PIs, up to at most 3 years of being part and of having this a permanent position or something similar to professor. They can still join as well. And then also here we have this very nice map, which was actually introduced by Alex, where we have now an overview of our community across the globe. And we actually recently had now also a member joining from Australia, which is super amazing. So we have only one continent left, and that is unfortunately still Africa. And I remember also talking about this already 2 years ago at the last IOL conference in Montreal, that we're still looking for people from there. So if you have my call to you, if you have anybody that you know who is actively working in research there, then of course approach them and invite them to join us. And I think it would be very nice to also enrich people, have people from Africa also be part of our network. It's super amazing. And I am just thinking widely, I would actually expect that there should be some people interested in Origins of Life. And you think about the greenstone belts in South Africa, or I don't know, we should be able to find someone. I'm convinced. Then with that, just some short updates about the website. So we have actually now finally a subsection on our website, which is targeted towards undergraduates. So would usually be people who are interested in origins of life, maybe some people in their bachelor studies, but they're not actively working yet in origins of life, so they cannot join, or at least can, of course, cannot join yet. But we, of course, also wanted to make them curious and like resources. So you will find there a whole list of different resources relating to origins of life, and also kind of some tips how to start to get into the very broad interdisciplinary field. And then, of course, if they then become, hopefully then, because of that, active in research, then, of course, they should join us as well. And as I wrote here on the right-hand side, if you have any ideas what to add to this website for undergraduates, just drop me an email or to the board, and we could also add, as we're always looking for. Yeah. Then there's also another thing that is also new since the last organization update, that we also have now a publication site where we have publications that emerged within the network. So I think it's also nice for people to see that when you take part in OoLEN, you might even have the chance to get a co-authorship, because people are actually discussing science, for example, in the internal discourse forum or on the conferences. And this already led now to 3 publications. The last paper, which is right now in the peer review stage, which was kind of led by Cole, where we have a nice overview over the methods and origins of life. And that's super exciting. And of course, we're always happy to contribute to these kind of things when you're part of the network. And then, as we are also here right now, literally we are streaming on YouTube. So we have now a YouTube channel, and we have these also, we have these recurring series of public seminars. So I know we are all very tired of this already, but you always have to say this in every YouTube video or stream. I invite you to like and subscribe, of course, to us. And so you will not miss out on any future meetings. And when I'm already doing this, I can also tell you that we also have a newsletter, which is also recurrently always showing you delivering you the newest juice from the network into your email inbox. So of course also subscribe to that. Then, of course, I want to just say a very big thank you to Yannick, Omer, and Celia again on behalf of the executive board, and also everyone in both the network. And big thank you for organizing this year's edition of our conference. And it's a super amazing experience. I'm super enjoying it, and it's really great thing to have in this pretty city of Strasbourg. And with that, I already wanted to introduce our invited speaker today. Sean McKayen will today talk to us about fossil microbes and the imposters. Sean is actually co-director of the UK Centre for Astrobiology, and he is also the PI of the Planetary Paleobiology group. That sounds really interdisciplinary. That sounds exciting. And yes, Sean, just take it away.
Thank you for that kind introduction. I guess I should share my screen, shouldn't I? Is that working all right? Can you see my slides okay?
Yes, looks good.
Okay, hopefully no irritating overlays or anything. Yeah, so thank you for having me along. It's a pleasure to talk to this audience. Hopefully this will be of interest. Anybody here who was at IANA or the Origins meeting in Cambridge the other week, I apologize for giving the same talk 3 times in a row, but it's quite a busy time of year with the start of semester here, so I'm afraid I need to do that. What I want to talk about today is the search for evidence of life in the form of fossilized microbial remains. So I want to start by saying some really positive things about fossil microorganisms. I'm going to be saying some more critical things about them later, but I want to make it really clear that I'm not in any way dismissing the importance of microbial paleontology, which is, after all, in many ways my own discipline. So let me emphasize that fossilized microorganisms are an extremely valuable record of the evolutionary history of life on Earth. And they are a primary source of information about the early evolution of eukaryotes, for example, as well as many other groups. There are well evidenced reports of fossilized archaeal cells in some settings, chiefly hydrothermal mineral veins. There are incredibly widespread and diverse and informative fossil cyanobacteria, these photosynthesizing prokaryotes that have transformed the Earth system over time by oxygenating the environment, and which in the fossil record often look very similar to cyanobacteria that we have in the world today. So you can go back 2 billion years and find beautifully preserved cyanobacteria in chert that look the same down to the patterns of cell division and the distribution of pigment in a colony as modern representatives of the same group. So cyanobacteria have the most amazing fossil record, but other groups of bacteria are represented as well. Iron oxidizing bacteria, which produce sheaths coated in iron oxides that preserve well. Sulfur oxidizing or sulfide oxidizing bacteria also make quite large tubes that preserve well in some settings. And many other bacteria are represented in the fossil record. We can't always tell what the organisms originally were, as in these pyrotized rods and coccoids here. Probably something heterotrophic, maybe sulfate-reducing bacteria. Not always easy to say because the morphology is quite simple. But what we do know is that the fossil record of microorganisms in general is a really important and valuable source of information about ancient life. And the preservation I'm talking about here is not just the morphology of the cellular remains, the sheaths and cells and colonies, but even sometimes the chemistry can be preserved in these kinds of fossils. And here I want to highlight a study that was led by Corentin Laurent in my group, published last year in Nature Communications, focused on one of the best-known fossil assemblages in Scotland, actually, the so-called Rhynie Chert of Aberdeenshire. So this is a silica deposit that formed about 400 million years ago, where a hot spring kind of geyser system was discharging silica-rich fluids onto the land, and the silica was entombing and permineralizing all kinds of organic remains, plants, fungi, arthropods, as well as many bacteria, and preserving them like insects in amber, and they've remained there for 400 million years, the organic remains of this biomass locked inside the silica matrix. And with infrared spectroscopy, you can analyze this carbonaceous material that these fossils are composed of and find the molecular fingerprints of the various functional groups and moieties inherited by this carbonaceous residue from the original biomass. So that's not to say that the original biopolymers have survived intact, far from it. They've very much been altered into kerogen, but there is molecular information surviving that tells you about the original organisms and even classifies them just with an unsupervised principal components analysis into eukaryotes and prokaryotes. So again, many of these are microbial fossils, microorganisms, and again, a rich source of information about ancient life. Why do we care about microbial fossils at all? One reason we care is that microbes are in many ways the dominant form of life on Earth. As I tell my students, it wasn't the dinosaurs that ruled the Earth. It's not the humans that rule the Earth now. It's always been the microbes. Ever since the origin of life, maybe 4 billion years ago, maybe you want to tell me a bit later or a bit earlier. I don't know, but for most of the history of life, it's just been microbes, the only game in town. And they've left behind not just a sort of cellular fossil record, but also other types of geochemical and geological record, like isotopic anomalies in carbon, iron, sulfur, and other isotope systems dating back to about 3.5 billion years. The famous offset in the carbon isotope record between organic and inorganic carbon, indicative of a global carbon cycle in which phototrophy is important. That offset goes back 3.5 billion years. The stromatolite record, many of you will know. Stromatolites are finely laminated, kind of convex, microbially produced sedimentary structures that fossilize very well, and again go back about 3.5 billion years. And then alongside those records, you also have the more conventional fossil record. But really, it's only a little over 500 million years ago that macroorganisms show up on the scene at all. So if you really want to understand the history of life on Earth, fossil microbes are an important resource. And they're also among the things that we hope we might one day be able to find on Mars. If you compare the timeline of early Martian history at the top of this slide with the timeline of early Earth history at the bottom of this slide, you can see that that period of Martian history characterized by lots of activity of liquid water at the Martian surface overlaps in time with some of these earliest forms of evidence for life on Earth. So when life first arose on Earth and became widespread on Earth. We're pretty sure that Mars was essentially a habitable planet at that time. There were rivers and lakes and even seas and so on. And they produce sedimentary deposits now being explored by rovers like Curiosity and Perseverance, and hopefully Rosalind Franklin in the near future. And in those deposits, we may find fossils of one kind or another. These rovers don't have the kind of microscope capability to actually see microscopic fossils, but it's hoped that maybe we'll find some when samples are brought back to Earth by the NASA ESA follow-on mission to Perseverance in the 2030s. And just to plug an old paper of mine, if you want to read more about the search for fossils on Mars, here's a review we wrote that goes through the various lithologies on the Martian surface that may contain fossils of one kind or another, and remarks on what the priorities should be. So I've made a case, I hope, that fossils of microbes are important. And now I'm going to raise what I think is also an important problem with the study of these fossils, and that's the problem of pseudofossils. Unfortunately, microorganisms are relatively simple in their morphology, and they overlap in their structural morphological complexity with a range of non-biological structures, abiotic structures, that can also be formed in nature and preserved in old rocks. Chemically as well, fossils tend not to be as chemically rich in complexity as living organisms. They're far simpler, and their chemical composition is in fact also kind of impinged upon by the chemical complexity that can develop abiotically. So this is our problem. How do we distinguish true fossils from abiotic mimics or imposters that might be similarly complex in their composition and in their morphology? And historically, this problem has been a real impediment to the progress of astrobiology, and in some ways also a stimulus to the progress of astrobiology. And a really famous example of that would be the story of Martian meteorite ALH 84001. Many of you will have heard of this. 1996, a paper was published by McKay and colleagues reporting what seemed to be multiple lines of evidence for the presence of ancient life on Mars captured in this meteorite that had found its way to Antarctica and been picked up by NASA scientists. And those lines of evidence included microscopic structures shown in the central image here that looked like fossilized microbial cells, as well as little carbonate mineral globules that were interpreted as the possible products of microbial carbon respiration, magnetite crystals that looked a bit like the magnetite crystals that magnetotactic bacteria can make, and even the presence of simple organic compounds, polyaromatic hydrocarbons. And it was argued that all of these lines of evidence together made what looked like a compelling case that this was evidence for early life on Mars. And I remember the president at the time of America, Bill Clinton, giving a talk on the lawn of the White House announcing this exciting discovery. Unfortunately, it turned out that that all of these supposed lines of evidence for life actually had simpler and more credible non-biological explanations, including these little structures that turned out to be a combination of mineral edge effects and sample coating artifacts for electron microscopy. Another famous debate about pseudofossils concerns a famous silica deposit, chert unit called the Apex chert from Western Australia, about 3.5 billion years old. In these cherts, a well-known paleontologist called Bill Schopf, who has made many great contributions to Precambrian paleontology, reported the discovery of microscopic carbonaceous filament structures, which he interpreted as fossilized cyanobacteria. You can see them illustrated on the left here in the figure from his original 1993 paper. So we have these kind of chains of carbonaceous residues that appear to be a bit sort of segmented, as if they were cells lined up in a filament or a cyanobacterial trichome. But this was quite quickly controversial, and the figure on the right here shows images taken by Martin Brazier and colleagues in Oxford and published in their Nature paper in 2002, showing that if you looked at the full diversity of the morphologies of these structures, rather than perhaps just cherry-picking the ones that seemed most lifelike, they actually ranged over at least a couple of orders of magnitude in size, and many of them didn't look at all like any known organism. So that was one problem with the idea that these were fossils. And then it turned out, when Dave Wasey and colleagues really studied these things using very high-resolution techniques, that although they do contain some carbon, these filament-like structures are actually made predominantly of clay minerals that are composed of these kind of phyllosilicate sheets oriented perpendicular to the length of the filament. So in fact, what these seem to be is delaminated clay booklets, particles of clay mineral sheets that have expanded and produced these accordion-like structures. And then the remaining porosity around those clay sheets has been filled in by carbon. So now we have an abiotic story to tell about how these structures originated. That's at least as consistent and maybe more consistent with all the evidence we have than the original biological interpretation. There's still some controversy about these. I'm not saying that they were definitely pseudofossils, but the case is much weaker than it was that they are fossils. Similarly, a few years ago, a team at UCL published the discovery of some intriguing little iron oxide tubules from a chert that might be as old as 4 billion years old, from a greenstone belt in Canada. And these iron oxide tubules, shown in this image on the left, which is just photographed, were morphologically quite similar to iron oxidizing bacteria, which produce sheaths coated in iron oxide. And they showed other curious morphological attributes, such as a tendency to radiate from knobs of hematite, which is an iron oxide mineral. And as this quote here shows, there were also tubes that had internal coiled, branched, and twisted filaments. So this complexity of the morphology reminded these authors strongly of biology, and in their view was evidence that these things were actually fossils of very ancient iron oxidizing bacteria. When I looked at these, I saw an alternative possibility because they reminded me not so much of fossils as of chemical gardens and the product of a chemical reaction or family of chemical reactions that's been studied for a few hundred years now, and which you can easily do in the lab. And if you do this reaction on the bench, you'll quite easily produce structures that indeed consist of hematite knobs with radiating filaments and tubes made of iron oxide, or if not hematite exactly, then a precursor to hematite that would be modified into hematite by the pressure and temperature conditions during burial. And these filaments that come out, have a similar size to these ancient examples, a similar looking cross-section and so on. So I argued that maybe these ancient structures are not fossil iron oxidizing bacteria, but actually chemical gardens or something maybe analogous to them, even though it's not entirely clear whether the conditions in which they formed are plausible chemically for this type of reaction to take place. It's at least worth considering as an alternative to the biological hypothesis. And we're continuing to do work exploring this. So my recent student, Melanie Podbielski, has submitted a paper which is now in review, comparing, using a range of different measurements, the tubes made by iron oxidizing bacteria and the tubes made by iron mineralizing chemical gardens. And I'm not even going to tell you which side of the screen is the chemical gardens and which side is the iron oxidizing bacteria. The point is they look very similar indeed. So these are just a couple of case studies that highlight a much more general problem, which is that many abiotic natural structures can mimic the simple morphologies and indeed chemistries that we associate with microbial fossils. And working with Julie Kosmidis at Oxford, I published a paper a few years ago reviewing all the ways in which natural processes that we know about might fool us in our search for fossils on Mars. And the images you see on this slide are taken from that paper. All of these things, I hope you will agree, look at least reminiscent of either organisms or fossil organisms, complex morphologies that look like dividing cells or filaments, chains of cells, branching structures and so on. But all of these are actually the product of chemical reactions or non-classical crystallization processes, either in the laboratory or in natural environments, analogous to environments that could have existed on early Mars. And here's the paper, if you'd like to take a look. Let's go a bit deeper now into pseudofossil land. I want to make a general point about astrobiology, which is that in whatever context we're searching for biosignatures, whether it's the atmospheres of exoplanets or the fossil record of early Mars, or the chemical complexity of fluids vented from Enceladus, or whatever it is, we're always basically faced with the same kind of problem, which is to discriminate the signal of life from some abiotic baseline that we may not know very much about. And if we can't discriminate the signal from the baseline, then we're going to make false positive errors where we think we've discovered life, and also false negative errors where we mistakenly think that we haven't discovered life, when in fact, that's exactly what we have measured. So when you think about things this way, you realize that actually exploring the abiotic baseline should be a really important and central empirical research program for astrobiology. In other words, we need to characterize the diversity of complex abiotic materials and structures that form in the environments that we're interested in. And that's what I've been trying to do in my research on fossil microbes for the last few years. So for the remainder of this talk, I want to focus on just one example of that, and that's the example of structures, complex lifelike structures found in agates. So in case you're not familiar with agate, this is a type of mineral deposit that forms inside cavities in rocks where the circulation of groundwater leaves behind a kind of residue of silica that accumulates over time and fills in these cavities. So if you have, for example, empty gas bubbles or vesicles in a basaltic lava flow, over time in the subsurface, circulating water will fill those vesicles with silica, and you'll get these kind of positive structures that often have really interesting mineral inclusions within them, made of clay minerals or metal oxides or oxyhydroxides. And these inclusion structures, entombed within the silica, can be really morphologically quite impressive. We don't know very much about the chemical conditions under which they form. People have tried to constrain the temperatures and so on, but it seems that the constraints are quite weak, and it could be anything from a couple of tens of degrees to hundreds of degrees Celsius. The silica itself eventually takes the form of quartz or mogonite, and the structures entombed within it, as I say, are generally composed of iron oxides or oxyhydroxides, or sometimes magnesium oxides or oxyhydroxides, or clay minerals, so aluminosilicate minerals. Here's what some of them look like. Hopefully you're impressed by these things. They look like feathers or trees or branches or fungal mycelia. These are different varieties of agate with structures more or less known to collectors. Very few geologists have actually studied these. The material you see at the bottom there is classified as a moss agate because the inclusion of iron oxide filaments connected in a network looks a bit like moss. Some of my colleagues think this is actually a fossilized, fungal mycelium. Others, like me, are not so sure and suspect that abiotic processes might be at play. People have realized for a very long time that agates are kind of natural laboratories for self-organization. We do know that many of these patterns are abiotic in origin, and there was classic work by Lisa Gang at the beginning of the 20th century to understand and also to reproduce in experiments some of the banding patterns that we find in agates. More recently, people have focused on the very fine scale alternating layers of different silica polymorphs that you see under the microscope in the silica part of the agate, as an example of a self-organized pattern that formed sort of spontaneously through the internal dynamics of the system. And people have also studied the formation of spiral patterns that look a bit like fossil snail shells that are found in some agates, and that seem to result from some kind of screw dislocation mechanism during the crystallization process. I've been working on agates from a variety of different localities over the last few years, and for the remainder of this talk, I want to focus on agate from just two localities. Chihuahua in Mexico. Here's a close-up image of this, a few centimeters across, showing some of the spectacular iron oxide inclusions present in this agate, and also the agates in the Pentland Hills that I can more or less see from my living room window in the south of Edinburgh. These are lava flows about 400 million years old, where again the vesicles in the lava flows filled with silica to form these agates. So the Edinburgh agates are much older than the Mexican ones by a factor of about 10. As soon as I started looking at these things under the microscope in specially prepared microscope slides, I noticed structures that in the past have confused people, and in the past have been mistaken for fossils, like these so-called ambient inclusion trails. So in the image on the left, you see an example from the Edinburgh agates. These are structures formed by a mineral grain, which you see as this little opaque speck at the end, with this kind of long tunnel behind it through the quartz of the agate. If you go back to 1923, you can find examples of people describing similar things from ancient silica deposits as fossilized algae. But it's been realized for a long time that these are the result of some unknown non-biological process through which these tiny little solid grains are propelled through the quartz medium over time, leaving a kind of tunnel behind them. We also, in these agates, found structures that, again, remind us of chemical gardens, so not only in the structure of the tube itself, but in the pattern of silica precipitation around them. So this is the graphical abstract from a paper written by my colleague, Pamela Knoll, and published in Earth and Space Chemistry a couple of years ago, where the rightmost figures here show structures in our agate, and the leftmost figures here show, what we can produce in the experiment, where we have a chemical garden filament of iron oxide with these kind of bubble-like structures of silica precipitated on it. So that's an interesting similarity. But the real surprise was the discovery of these microscopic spider-like structures seen on the left-hand side of this slide. These examples are from the Edinburgh agates. They are hundreds of microns in characteristic dimension. They're composed of hundreds of radiating filaments surrounding a central core, a little bit like those structures that Todd et al. Described as fossil iron oxide in bacteria. And the radiating filaments also branch in a rather interesting way, and they're present both in the Mexican and in the Edinburgh agates. The first thing they might remind you of would be the kind of fungal contamination you sometimes see in microbial growth media if you're a microbiologist, where you get these radiating kind of clots of fungal hyphae. Or if you're an experimentalist working on silica carbonate biomorph systems, you might have seen structures that look a bit like this as a result of that famous non-classical crystallization pathway. Here are some more close-up images. You can see that many of these filaments have a really interestingly sinuous curving trajectory through space. Many of them also branch. So here are some examples from the Mexican agates showing 1, 2, 3 orders of Y branching, side branching as well, and sometimes even anastomosis, the reconnection of independent branches. And the reason I emphasize all these morphological properties is that they are often discussed in the paleontology literature as things that you can use to identify true fossil microbes that you don't expect non-biological imposters to be able to do. But what are these structures in my agates? Well, one thing that's a really important clue is that these filaments are crystalline. They have a nice faceted polygonal cross-section, nice little hexagons that you see under the electron microscope. So in fact, these are crystal structures. They're not hollow tubes or anything like that, for example. And they're quite similar to things that have been described before from hydrothermal calcite veins, where similar looking filaments of pyrite, not quite as impressive morphologically and not radiating from cores, but nevertheless overall quite similar, have been described. Another thing we can do with the electron microscope is get some preliminary kind of elemental characterization, and what we find is that while the filaments are made of iron oxides, and we confirm that with Raman spectroscopy as well, goethite and hematite, the centers of the cores from which the filaments radiate are actually slightly enriched in copper and sulfur and even a little bit of carbon. Interestingly, and you can see that in these element maps here, this little bright spot of copper and sulfur just at the core of these kind of spider-like radiating structures. So the challenge now is to understand how these things actually formed, and we have a working hypothesis which is a little bit early to be sharing with you all, but maybe you'll have some thoughts on it. Our working hypothesis is that these things originated as tiny grains of a mineral like chalcopyrite, a copper iron sulfide mineral that's commonly found as an inclusion in basalt. And if we had an aqueous solution or silica gel inside these cavities in the basalt that later filled with silica, and you had little grains of chalcopyrite sitting in that space, they may well have gradually oxidized. I mean, the stuff is now full of iron oxide, so there was some process of oxidation. Now, when you oxidize chalcopyrite, the insoluble products of that tend to be hematite and goethite. There's experimental literature on that. So in other words, the copper and sulfur tend to leach away into solution, and you just produce these iron oxide minerals that could have formed a kind of rusty coating on the original grains. So how do we get these filaments? Our hypothesis is that as this rusty coating of iron oxides grew, you formed crystals, for example, of goethite, that were better able to adsorb silica on some crystal faces than on others. And that led to the smothering of some crystal faces by silica, for instance, those that had more edge sites for adsorption, while other crystal faces, maybe just one crystal face, the 001 direction, for example, in goethite, was able to continue growing because it wasn't being smothered by silica. And in that way, you can get a kind of one-dimensional crystal growth. So you end up with quite a linear crystal growth process. So you can imagine these crystals growing just in one dimension while being coated with silica, and you can see that where you get gaps in that coating of silica on the sides, you could have side branches forming in those gaps. And where you get some occlusion of the end of the growing crystal by a little bit of silica, that could also be the cause of a Y-type branching point. So this is a bit speculative, but this is one possible mechanism that could generate not only these filaments, but it could also explain the branching patterns that we see. And of course, what we really want to do now is explore these kinds of processes experimentally and see if we can verify any of this. So I'll leave it there. That's just a kind of snapshot of where we are with this project. And I'll put my summary slide up. Summarize: microbial fossils are an important archive of early life on Earth and a valid target in the search for life on Mars. But there is this pseudofossil problem. I think if you're interested in morphological complexity in the abartic baseline, agates are a really interesting material to investigate. One thing I forgot to say, of course, is that agate is also a very Mars-relevant material. There's plenty of vesicular basalt on Mars. There's also plenty of aqueously deposited hydrated silica. So it's just a matter of time before somebody finds an agate on Mars. Plenty of iron oxides, of course, as well. And as I've shown in this talk, we do find interesting new varieties of biomorphs, pseudofossils, in agates. In this case, from two different continents separated by 300 million years, which indicates to me that these are probably actually very widespread. And on the right-hand side of the slide are members of my group that I should thank who've been involved in this work: Pamela Knoll, Corentin Laurent, Eleni Podbielski, Georgia Brown, Edwin Rodriguez-Joule, and Mia Frothingham. And I've thanked some funders at the bottom there. And the last thing I'll say is that in March, I expect to be able to share an advert for a new postdoc position to work on microbial fossilization. So please email me if you want to get your foot in the door and let me know that you might be interested in that. And please share that if you know anybody else who might be interested. Thank you very much.
All right. Thank you, Sean, for this very interesting talk. I guess I will start with the first question, if you don't mind. So on the slide where you were showing the fossils of microbes, archaea, cyanobacteria, and so on, I noticed that most of them, I think, were filamentous organisms, so they formed filaments. Does this, forming filaments, does this help actually with the fossilization? And would it have advantage, for example, over the biofilm forming organisms or free living microbes?
Thanks. That's a really interesting question. I think when it comes to cyanobacteria, we have quite a diversity of forms, some filamentous, some not. And what's thought to explain the abundance of fossil cyanobacteria is the fact that the polysaccharide-rich sheaths that these bacteria secrete seem to have a good preservation potential. They seem to resist decay quite effectively compared to just your regular peptidoglycan bacterial cell wall, for example. Cyanobacteria also are morphologically interesting enough that we can usually, or at least sometimes, figure out what type of cyanobacterium we're looking at, which is a very helpful thing to be able to do. Whereas for many other types of organisms, okay, they might make coccoids or rods or filaments, but they're not morphologically complex enough to be able to say really anything about the classification of those ancient organisms. Iron oxidizing bacteria, we know that some of them also make filaments that have a good preservation potential just because they kind of fossilize themselves while they're still alive and get coated in these iron oxides. Other types of microorganisms, I don't think there's anything necessarily special about being a filament, except that it's easier to recognize in the rock record than just microscopic micron-sized kind of blobs, which do sometimes get interpreted quite dubiously as fossils. With filaments, I think people see that shape and are more naturally drawn to the biological conclusion. Although really, it's such a simple shape that maybe they shouldn't be. Have I answered the question? I'm not sure.
Yes, you did.
Okay, thanks.
Yes, thank you. Does anyone in the audience have questions? Amit?
Yeah, hi. I wanted to ask about copy number. So you might find a complex morphology in a mineral and then think that it's like abiotic or like it's amorphous. It's hard to determine whether it's abiotic or biotic origin. But then if you find 100 copies of it or like 1,000 copies of it, would it be considered like more likely to be biological because of the, it's like non-trivial to find so many copies of it?
Yeah.
Or how would you interpret that?
So I guess when we interpret one thing as a copy of another thing, what we're saying is that they're similar enough that we consider them to be essentially the result of an equivalent process. And I guess if I'm understanding your question rightly, what you're kind of hinting at is that in a population of cells, you would expect them all to look maybe more similar to each other in their size and in their aspect ratio and so on, than a population of non-biologically formed chemical precipitates or something like that. Am I getting your question right?
Yeah, that's my intuition.
Yeah, so that's a widely shared intuition that's been discussed for a long time. It used to be argued that if you had a size distribution of microstructures that looked like a normal distribution, that was good evidence that it could be a biological population. And then, of course, people realize that actually lots of things in nature give you a normal distribution, and that's not actually a particularly robust line of evidence of biogenicity. It is true that if you have a wildly varying characteristics size of your structures, like for instance, those filaments in the apex chert that seem to vary over at least 2 orders of magnitude in size, that doesn't sound like at least it doesn't sound like it's a population of the same organism. And you could have mixed populations of different organisms that happen to be different sizes. But then you might expect a kind of multimodal distribution of sizes. But then again, you could have multiple abiotic processes giving you a multimodal distribution of sizes. Similarly, if you think about these spider-like structures in the agates, the distribution of diameters of those filaments is quite tight. They're pretty much all around 3 microns roughly, which is maybe the same as what you would expect for certain filamentous microorganisms. So I'm sympathetic to the general intuition that we should be measuring this kind of thing, and we should be looking at distributions across populations and not just the shapes of individual objects. But in practice, it's still often quite hard to infer whether something's biological or not from these kinds of statistics. I hope that answers the question.
Yeah, definitely.
Okay, does anyone else in the audience have a question? Raise your hand or just speak up. Okay, let me quickly check the YouTube chat for questions. Okay. So if no one else have any more questions, then, well, I actually have one more, but I'm not sure if I should ask it. I don't know if it's even a valid question. Anyway, I'll ask. So all these structures, chemical structures that people see, and we as humans tend to misinterpret them as biological structures. Why do you think when we look at something, for example, round shaped one is like several balls stacked together under a microscope, why do we immediately go to think that it is something that is alive?
I think because we're making a kind of induction based on our experience. Most of the time when we've seen that kind of shape before, it was in a biology classroom or something like that. So that's maybe part of the answer, that we've been trained to see certain shapes as biological. I have a hypothesis that if anybody knows a psychologist who'd be interested in thinking about this, I have a hypothesis that we also have a deeper intuition, that is maybe a kind of something that we maybe have from a very young age, that maybe people have always had, that if a shape is sinuous or branching or has certain other characteristics, it just looks biological to us more than a shape that's kind of crystalline or hard-edged or simple. My guess is that that's actually a deep-rooted psychological kind of shortcut that we maybe all take, and it would be really interesting to work with a psychologist to just survey lots of people out there and see if people tend to classify shapes the same way as each other and as scientists do. But as far as I know, that's not been done.
Yeah, I think you might be onto something here. I think I read something, it was from the field of linguistics, but people tend to also classify words as sharper shape or softer shape. So I think we have some kind of innate mapping.
Yeah, I read about that too. Yeah, that's an interesting
Yeah.
parallel. Yeah.
So you definitely might be onto something there. So yeah, if you get a psychologist to collaborate, that would be a really cool study, I think. Okay, so I am assuming no one else has questions since no one raised a hand or was speaking up so far. Well, okay. Then thank you, Sean, so much for joining us today and giving us this interesting talk. Thank you, everyone, for attending and everyone who's watching or will be watching in the future this recording. And stay tuned, and we will see you next time. Thank you very much.
Thanks, folks. Bye for now.
Bye.
Thank you.